Today I will be presenting to you the results of my master's thesis research titled Olive Ridley Sea Turtle Embryo Mortality as a Function of the Nest Microbial Community at OSTIONAL, Costa Rica. The olive Ridley sea turtle is listed as endangered and threatened throughout its range. This species of sea turtle has nesting sites along the Pacific, Atlantic, and Indian Ocean, and exhibits a unique mass nesting behavior at few of these nesting sites. My study site is located at the OSTIONAL Wildlife Refuge on the northern Pacific coast of Costa Rica. Here, mass nesting events estimated at up to 500,000 nesting females over a period of only 7 days. This behavior is believed to be a predator satiation technique in which the nesting females overwhelm the large population of predators with an abundance of eggs. Despite this large population of nesting females, concern remains that the low hatching success at this beach is not enough to sustain the population over the long term. Several previous studies propose that sea turtle embryo mortality is associated with the high microbial load that likely results from this large input of organic matter. However, no previous research has directly quantified microbial abundance in the nest sand and its direct and/or indirect effects on hatching success in an in situ environment. Microbes, such as bacteria and fungi, compete for the same resources as the sea turtle embryo. Microbes decompose organic matter and consume oxygen and release metabolic heat in the process of doing so. Similarly, the sea turtle embryo requires oxygen for development and develops under very specific temperature requirements. To better understand how these two interact, let's look at the cross-section of a sea turtle nest. At a typical sea turtle nesting beach, there's relatively low amount of organic matter in the nest sand. At a beach such as OSTIONAL, however, the large input of organic matter may be driving an elevated rate of microbial activity. In this case, the microbes are consuming so much oxygen that perhaps they are disrupting the diffusion of oxygen to the outside of the nest and adding metabolic heat to the nest temperature. In this case, it's possible that the microbes may be favored over the developing sea turtle embryo. This leads me to my main hypothesis that as microbial abundance increases, hatching success decreases due to this competition for resources. The primary objective of my study was to determine whether a relationship exists between sea turtle hatching success and the microbial abundance of nest sand. And second, determine whether any indirect effects of microbial abundance affect hatching success. I did this by relocating nests into experimentally treated sand, and I quantified microbial abundance using molecular techniques. I also monitored various variables that are relevant to both microbial decomposition and the developing sea turtle embryo. My experimental design used a hatchery-like setup of experimental treatments into which sea turtle nests were relocated. We had two different types of treatments. The first, the soak treatments, involved the removal of sand, a soak in a treatment for 24 hours, a rinse, and the replacement of sand. We used an antimicrobial treatment of a solution of bleach. We also soaked sand in fresh water, and we also included a control for the removal and replacement of sand alone. Topical treatments, on the other hand, involved the topical application of either seawater or fresh water to an experimental treatment plot. We also included treatments in which there was no treatment whatsoever to the sand. All of these were replicated 15 times, and we left 5 of these with no nest in order to control background levels. For the purposes of this presentation, I have grouped together the treatments for the soak and topical treatments. I have also left separately, in blue, the control treatments. And just to reinforce the difference between these two types of treatments, again, the soak treatments involved a disturbance of sand, whereas the topical treatments did not involve any disturbance whatsoever, and you'll see that this comes into play later on. We relocated all of these nests within the first 2 consecutive nights of an arrabata and placed 100 eggs into each plot. We also included a temperature data logger and tubing to measure the partial pressure of oxygen throughout the incubation period. We collected nest sand at the time of excavation, and from that sand we isolated microbial DNA, which we then quantified using quantitative polymerase chain reactions. We used universal primers that targeted the 16S and 18S rRNA gene. This allowed us to have a quantified copy number per gram of nest sand, which we can use as a proxy for overall bacterial and fungal abundance. As you can see, this was a whole lot of data, and today I'll just be presenting to you the factors that we found most relevant to determining hatching success at our beach. Here you'll see the treatments across the bottom axis along with the treatment type, and hatching success along the y-axis. There was a significant effect of treatment on hatching success, with the treatment of removal and replacement of sand only being the most successful at increasing hatching success. When we separate these out just by treatment type, we found that all of the treatments that involved a disturbance of sand had significantly higher hatching success in comparison to topical treatments. Here you'll see nest oxygen content along the y-axis with the incubation period along the bottom axis. We found that oxygen levels in all of the soak treatments were relatively stable throughout the first half of the incubation period and decreased drastically in the second half of the incubation period. This is typical of a sea turtle nest, as the sea turtle embryo consumes relatively low amounts of oxygen, and it's not until the second half of the incubation period, when metabolism picks up, that oxygen consumption rates also increase. However, when we compare this to topical treatments, these treatments had significantly lower oxygen levels throughout the incubation period, and particularly in that first half of the incubation period. This difference in oxygen, particularly in the first half of the incubation period, is indicative of elevated microbial respiration rates. Here you see fungal abundance across the bottom axis on a log scale and hatching success along the y-axis. We found a significant effect of fungal abundance on hatching success. This correlation was highly significant and negative. However, when you compare this to bacterial abundance here across the bottom axis, there was a marginal negative correlation between these two factors. And you'll notice that this slope is not as negative as the previous fungal abundance effect on hatching success slope. So how do microbes affect nest oxygen and temperature? We found that overall microbial abundance was negatively correlated with the oxygen content in the nest, and positively correlated with nest temperature. So just as we expected, as you increase microbial abundance, there's a decrease in nest oxygen and an increase in temperature. And what factors are most important in determining hatching success? Our study found that the single most important factor in determining hatching success was the oxygen content of the nest in the first half of the incubation period, followed by an interaction between fungal abundance and the organic matter content in nest sand, as well as oxygen in the first half of the incubation period. This suggests that the fungal decomposition of organic matter is altering the oxygen levels in the nests and therefore negatively affecting hatching success. It seems that fungi may play a larger role than bacteria in altering the nest environment. And again, the removal and replacement of sand alone was the most effective treatment at improving hatching success for our study. This study has implications for conservation on both a small scale and a large scale. Present sea turtle management practices are becoming more and more dependent on the relocation of nests into hatcheries to protect nests from erosions and predation and poaching. Hatcheries tend to have more issues with organic loading as well as microbial infestations. On a larger scale, beach renourishment and wastewater management issues are also becoming more important, as is the anthropogenic input of organic matter. It's important for us to consider the quality of beach sand and the impact that this might have on the reproductive success of sea turtles at nesting beaches around the world. I'd like to thank my main sources of funding, primarily National Geographic, the National Science Foundation, and the Patty Foundation, all of which provided a majority of the funds for this research. Furthermore, I have many people to thank, many volunteers and friends that contributed to the success of this project, particularly my committee, all of my undergraduate lab assistants and research assistants both in the field and in the lab, as well as the sea turtle community for providing valuable input and advice for the entire process of this research. Thank you very much.